Okay, we're live.
We are. Okay, shall we launch?
Go ahead.
Okay. Hello and welcome everybody to Science on Screen. My name is Susan Whitehead and I'm here because I'm a huge, longtime fan of the Coolidge Corner Theatre and of Science on Screen. And I feel that I've been eyewitness to a lot of the discovery that was chronicled in From Controversy to Cure. So this wonderful series, Science on Screen, is made possible at the Coolidge through a major grant from the Alfred P. Sloan Foundation, as well as significant support from Reuben Anders Scientific, Ellen Hoffman, and Ken Loveday. And special thanks should go to the founder of this series, Richard Anders. So Science on Screen, as probably many of you know, because you've been to other of these panels, is a gem of the Coolidge Theatre, and it's an original of the Coolidge Theatre. And the intent is to tell the stories and the histories behind scientific discovery. Now, this story, From Controversy to Cure, is particularly relevant for all of us because it touches all of our lives individually. Not all scientific discovery touches us all so personally and so deeply, but this tells the story of the revolution in molecular biology and the understanding of basic biological processes and how that's been translated into cures and therapeutics and things that have a very positive impact on all of our lives. So this story, this film tells the story of the people and the forces behind discovery. What makes it so interesting for our community in the Boston area is everybody who you see on this screen. These are all your neighbors and community members and people who have lived in this community and made these major scientific discoveries as members of our community in this area. This is a story born out of passion for the discoverers and also born out of passion for the filmmakers. So I want to just give a special shout out to the people who created this film. You can see one of them on screen, Joe McMaster, the director. Larry Gallagher has also joined us online, and Jean Dunoyer is also online with us this evening. So to introduce the panel, I'd like to introduce Antonio Regaldo, and I think Antonio is also online and visible to you. And Antonio is the senior editor of biomedicine at MIT Technology Review, and he'll be moderating this panel this evening. So welcome, sit back, enjoy yourselves. It's a great story. Antonio, over to you.
Thanks, Susan. So I guess in a way, this is a discussion about a place that I work, Kendall Square. The film Controversy Cure calls it, the biotechnology capital of the world, less than a square mile around MIT, 100 biotech companies, many of the largest pharmaceutical firms, and as Eric Lander, who's not with us, says in the movie, he calls it the densest collection of scientists in the known universe. And actually, sometimes when I have guests that come to visit me, I take them out to the median in Main Street, which is between the Cancer Center, where Phil Sharp, I think, has his office somewhere in that building, the Whitehead Institute, and the Broad, and I can find the physical place that is that center of the biotech universe. So it's kind of ironic. It's a story about a place and about molecular biology, and we can't be in that place because of a pandemic, which potentially molecular biology and in particular companies in the region may solve. So just briefly introducing who we have, some of the creators or founders, you could say, of the biotech cluster in Kendall Square. David Baltimore, who's the founding director of the Whitehead Institute, a Nobel laureate, former professor at MIT. Harvey Lodish, hi Harvey, MIT professor of biology, co-founder of Genzyme. Nancy Hopkins, emeritus professor of biology at MIT. Sangeeta Bhatia, who is at MIT. She's a professor in the electrical engineering department and computer science. And finally, Joe McMaster, who's actually made the film that we're going to be talking about, From Controversy to Cure. So Joe, I just wanted to hand it over to you and wanted to hear your definition of what Kendall Square is and why it was that you decided to make a movie about it. Joe,
You're
Muted.
You're muted. Great.
There you go.
Is anybody else getting feedback from another audio? Okay, great. So it's just me. Not sure why that is. Anyway, thanks very much for joining us, and thanks to everyone on the panel here who could join us. I'd just like to quickly acknowledge all the people who gave their time to this project, including those you see here. And as well as all the many people who sat down for an interview with me, our informal group of advisors, including Susan Whitehead, Larry Gallagher, and my colleague Jean Dunoyer, all my colleagues at MIT, as well as Neil and Jane Papalardo and the Sharp Charitable Trust for making this possible. So at any rate, with that said, our aim in making this was really to explore the history of this extraordinary ecosystem of Kendall Square, and the kind of genesis of it. It was, in a sense, you know, if you spend any time in Kendall Square, you have this funny experience of one day there's a building there, and then a day or so later it might be gone and there's a hole in the ground, and then a week later there's a steel frame rising out of that hole, and it's just an extraordinary boom that's changing the landscape, and has been for quite some time. And we set out to sort of explore the question of why that is, what's happening there. If you ask people why Kendall Square is such a boom town, they give you two answers usually, I find: MIT and biotech. And so that was kind of the genesis of the film. We set out to explore just what it was that gave rise to the modern Kendall Square. And it turns out, of course, Kendall Square has a very long history that goes back centuries as a manufacturing hub. But the modern history really begins, I think, with a lot of the folks that you see on screen here, and the birth of molecular biology, as was said earlier. And there is no one better qualified to tell that story than the folks you see here. And I think they were the ones that were best able to answer what happened in Kendall Square. You know, why is biotech, why is it the so-called biotech capital of the world, which I'm sure is a disputed title, but nonetheless.
I think it might be still disputed by San Francisco. They have their own supercluster, Silicon Valley, and there's a long tradition of people trying to replicate these zones. You know, they'll put up a technology park or an innovation park, and it never works because people don't know exactly what ingredients and in what order. But I think with that, I would ask David Baltimore. I think the story kind of in the film, it kind of starts with your arrival. Is it 1960?
Yep.
At MIT, a school with no reputation in biology, no medical school. And I understand from the film that you appear in your sandals, and you know, and you're also an unlikely character to, I mean, a hippie, as it were, and also maybe an unlikely character to start this off. So, what was the unifying concept or the seed that was planted back then that grew into the Kendall we know today?
There are a couple of seeds. One was that MIT didn't have a traditional biology department. And so when the wise heads at MIT decided that they had to become involved in contemporary biology, they were smart enough to say, we missed all the past, let's just focus on the future. And the kind of people who did that were Salvador Luria more than anyone else. So I graduated from Swarthmore College. During that time, I spent a summer at Cold Spring Harbor, and I met Salvador Luria. And he said, I'm going to MIT. I'm going to start up molecular biology. Do you want to come? Well, the amazing thing is there was nowhere else to go. There weren't any departments of molecular biology in 1960. And so I was just overjoyed to know that he was going there and that this was going to be a center, and it became probably the greatest biology department in the country. And it was this focus on the future, always one step ahead. When Luria started working on bacteria and bacteriophage, that was the start of molecular biology. But he didn't stop there. He went on to say, we've got to work on animal viruses. We've got to work on animal cells. And he brought the generations of people, some of whom you see here, Harvey Lodish, Phil Sharp, Nancy Hopkins, and many others. And I can go on for hours naming them all. And that's what gave the impetus. But there was something else, and it was the spirit of entrepreneurship. Most of us, maybe all of us, grew up in seeing biology, molecular biology, as a very beautiful construction around the deep questions of what is life. It wasn't about making drugs. It wasn't about solving medical problems. We all suspected that that was in the future, but that's not why we were doing it. And so it was this aggregation of people who cared deeply about the structure of biological systems, wanted to know how DNA was able to control all the details of us and every other organism on Earth. That's a miraculous ability. And we lived through the period in which that was understood, and each of us made our own contributions to how it was understood. It's not an accident that all of this took place, but it certainly is an accident that all of us who were involved saw it and decided to make it part of our future.
The film makes the point that at MIT there was a tradition of entrepreneurship, of taking science and turning it into practical technology from the electrical engineering side or mechanical engineering, and that was easily, people who are in the biology department could easily do the same thing. But I wanted to ask Harvey Lodish, because there's some remarks in the film about when you started the biotech company, or helped start the biotech company Genzyme, that people kind of threw up their hands and wondered why it was anybody who was interested in these beautiful mysteries of life would possibly want to get involved in a company. So can you speak to how it was that the entrepreneurship component came in?
Well, again, it's an intersection, I think, of two histories. One is the history of entrepreneurship among MIT faculty. Among others, my brother Leonard was a PhD student at the Sloan School in the late 60s, and he was a marketing student, but started a company with his professor that did very well in a very different area. So we had a tradition of faculty helping taking research out of their own laboratories or offices into the community. But it was really the recombinant DNA revolution. And that's certainly described quite well in the movie. The ability for the first time to not just isolate genes that encode specific proteins, but to actually manufacture these proteins and use them to replace the proteins that aren't made in people with certain genetic disorders. For instance, diabetics who can't make insulin. Genzyme started really the vision of Henry Termeer, that there were rare diseases, such as Gaucher disease, in which a particular gene was defective and a particular enzyme was missing from the body. And as a result, something accumulated that caused problems, in this case, a certain fatty substance. And the realization that we could now manufacture this missing protein and administer it to patients, and really for the first time cure a whole variety of diseases that we couldn't cure before. And Biogen was started on essentially the same premise with interferon and others. Phil can talk about it. And other companies were started to make yet other biological hormones, other kinds of molecules. So a lot of it was really dependent on the technology that was being developed. And of course, MIT was a leader in the development of that technology as well.
Phil, what was your, I mean, you were founder of Biogen, also probably some other companies, Alnylam, I guess is one of them. How do you think that that the concept of entrepreneurship has evolved since those early days? I mean, it seems like it's reaching quite a fever pitch. I mean, what is your experience?
So I just, again, picturing MIT in this, my first interaction was a call from an ex-MIT alum who called me and said, Ray Schaefer, that he was part of a venture capital group. He was interested in me being a consultant for what became Genentech, or was Genentech, and an investment there. And then with him and Kevin Landry, who was a TA associate partner, they worked with Wally Gilbert and myself, and then a number of European company colleagues to start Biogen. And it was originally in Europe, and then came to Kendall Square in the early 80s. But I want to build on that comment that David made, that the basic science was the motivator for most of the work that was done at MIT and elsewhere. But there was this vision that this whole technology was going to create a whole new industry in plants, food, chemicals, and life and medicine. So it was the interest in taking the science and technology into the private sector to allow it to grow and become professional. That was the major motivator to get involved. So that was the baseline motivation for why Biogen was created. And then it evolved into the company that still is fabulously successful and doing wonderful things. Hopefully, they'll get approval for the first treatment for Alzheimer's. So it's a story of discovery being translated into a private sector that's had an enormous impact in the world. And much of that impact was anticipated in the early days of recombinant DNA. If you read the literature, you'll see in articles about the impact of this whole technology.
I appreciate that comment, because as a journalist, we always want to use the word first this, first that, first time. But if you're careful, you'll always see that someone thought of it before, often someone at MIT, and they wrote it down in this period. I wanted to take your point, also building off of David's point about the basic science. In the film, Nancy, you make this interesting comment about climbing Everest. I think that was you, is that right?
I think that was Susan Hockfield at the end.
Oh, was it
perhaps.
Susan Hockfield? Ah, okay.
But she can still answer the question.
You can still answer the question. I mean, to me, one of the exciting parts, you can't discover the South Pole anymore. You can't be the first person on the moon. There's no new shores to land on, but there's still, the telescopes and the microscopes. So the discovery is, if you want to reach a new place, you're going to go into this microscopic world. So Nancy, I wonder if you have thoughts about who is coming into biotech and biology now? I mean, what motivates people? And is it the smartest people in the world that want to be biologists, or do they go to finance? I mean, who is the group that is coming to biology these days? I'm sure they ask your advice. Unmute yourself, please. You're muted.
Sorry, my husband was making a lot of noise next door, so I had to mute myself. I mean, I'm probably not the right person to ask for two reasons.
Okay.
Just because I'm retired, and second of all, because I think about this a lot, actually, but I don't have an answer. So I'm hoping somebody else on the panel will. But because something David said also, I mean, the science when we started was beyond breathtaking. It was really was the secret of life. And so at the time that I started, I discovered this field in 1963, taking a class from Jim Watson at Harvard. So the science was absolutely breathtaking. And after 1 hour, I was a junior in college, 1 hour of Jim Watson, and I said, my God, that science is one day going to cure every disease. Okay, so that was sort of clear. But the thing that I thought at that time, 1963, but it's going to take 200 years before it's going to happen, but it will one day do that for us. And so I was captivated by it. But then, and I went into the field, was drawn into it, but it was the basic science that I really then fell in love with. Yes, it was going to cure every disease one day, but it was the passion for this incredible discoveries that were made about what life actually is, and there was this potential down the road. What's remarkable when I look back on it is what I thought would take 200 years was compressed into, I don't know, 10, 20. I mean, it went so fast. And so when I wanted to work on cancer, I actually wanted to work on cancer from the time I started. I didn't think it was possible. And by the time I finished graduate school working on bacterial viruses, I said, oh, now we can work on cancer partly because of David's work, reverse transcriptase, because viruses cause cancer. We understand viruses now. We can actually work on cancer. So in this very short period of time, suddenly you could actually work on the molecular biology of cancer, which was astonishing. But I think it was still, to me, very basic science because we knew zero about cancer at the molecular level when David opened the Cancer Center with Salva. I mean, we knew zero. Don't you think? Is that fair, David? Or did we know something?
Absolutely, we knew zero.
We knew zero. I mean, it was unbelievable. And so they opened the Cancer Center. And again, it just went amazingly fast. But it really was, as Harvey said, this recombinant DNA technology that came in and shortened the 200 years down to the 20 or so that we saw happen. So that was sort of how I saw it. I saw it as this extraordinary basic science that drew people like Salva and David, myself, Phil, Harvey, in with the hope, but not the expectation that in our lifetime, this could actually happen.
And Phil talks a lot about the commercial side of the revolution, but when Phil started off, it was the basic science that got him. I remember him so well.
I know you heard me, but you have to realize, David, I came out of a chemistry department at the University of Illinois. And in chemistry, this tradition of science to industry has been very old. 3 out of every 4 graduates from that institution went into the private sector. So I actually viewed a great deal of my approach to biology as bringing chemistry to it, and physical chemistry in particular. And I understood, or appreciated, let's put it that way. Yeah, everybody understands it, but I appreciated the impact of taking this science into the private sector and what it would mean. It took a long time for it to be developed. Nancy, it might seem like a blink of an eye to you, but decades were spent
Well, for each of us, it was a lifetime.
What?
For each of us, it was a lifetime.
It was a lifetime. And it's been, just exciting as it could be. And it's even growing in power, and its full impact we won't see for another several decades, if not another several lifetimes.
Right.
One thing,
Go ahead, Nancy.
Please.
Here's a controversy that the film could have been about, maybe, that it wasn't about Joe. I mean, the controversy was over the recombinant DNA debate, which was a big historical event, of course, a major thing. But there was this debate, David, I'm sure you remember, of even working on cancer. These people who were really basic scientists thought working on cancer was too applied. We should all be working still on yeast and phage and bacteria. I remember these arguments within the department. So it was a big step even to build a cancer center. And then the whole question of whether or not people should work in industry. Even though MIT had this great tradition, there was a controversy within the department between those people who thought it should remain basic and those people who saw the potential of the application.
Yeah, I was struck in the film, or in the interviews anyway. I know a number of you talked about how it was sort of seen as kind of unseemly for biologists to turn to industry, whereas, as Phil mentions, this had been going on in many other industries for a long time. What was that about?
Can I add another piece to it? I was transformed around 1970 by having a sabbatical visitor in my lab, David Nathan. It was the time I shared a lab with David Baltimore, if you'll remember.
Yep.
He was the head of hematology at Boston Children's Hospital. My lab was studying fundamental aspects of how red cells are formed. But together we began work on a genetic disorder of red cells. And I became incredibly impressed by the opportunities to first understand and then eventually cure medical diseases. I mean, a lot of medicine was opaque at the time. And they had no idea of what the underlying cause. Infectious diseases, yes, but cancer was not the only one. Many anemias that we were interested in, we had no idea what caused them. And what we created was also a large group of MDs who trained in our labs. All of us have had MDs in our labs who have gone back to research hospitals and medical schools and made fundamental insights in human disease. So I think it's deeper than simply saying, yeah, we took an idea from our lab and took it into the marketplace. We were really solving problems of disease. And you can't cure a disease until you understand it. And certainly the most recent discoveries, jumping ahead to the current gene therapies, that came out of many, many years of understanding the basic science, not just the basic science of how DNA works, but understanding exactly what is wrong in patients with a whole variety of disorders. And I think that also underlies a lot of our motivations. It certainly underlies my motivations for going into the business.
So one of the things that would be interesting, there is someone among us who's contemporarily starting companies, Sangeeta, out of an engineering school and the Koch Institute. And it would be interesting to hear Sangeeta talk about the motivation of someone now starting companies.
Yeah.
And actually, let me take the opportunity to also give you a related question from a viewer, Emma Dawson, who wants to know how the involvement in biotech companies affects how the lab does science. She wants to know, what is the backwards coming effect when you're starting companies?
Yeah, maybe I can weave that into my comments a little bit. So just to say that I arrived at MIT in the 90s, and this group on the screen are my heroes, just amazing foundations of the whole industry that you see today. And I think there are at least for engineers who are interested in applied solving problems of society, that's our mission. There was at least 4 other elements around the biology department, I think, that came into Kendall Square. So one of them you alluded to early on, which is engineering, and just very specifically chemical engineering was really part and parcel of the biotech industry making medicines. Those bioreactors were classical chemical engineering problems. And electrical engineering, so fabrication on the one hand and optics on the other hand, that was the way we got sequencing done. Microcapillary electrophoresis, those are engineering solutions, as well as chemistry solutions. So there was engineering. Then there was medicine, which Harvey alluded to, and MIT is situated right just over the bridge from MGH. And then when I came as a student in the 90s, and we still have today, there's a bus, it's called the M2 bus, and it goes back and forth to the Longwood Medical Campus of Harvard. And I spent many a day on that bus going back and forth. And so there really was this sort of connection between the biology and the medicine and the engineering. And then around it, we had our amazing technology licensing office. So you showed Lita in the film. And MIT, I think, was very mission driven in its view that it should get the inventions out there. That was an enlightened position that not all universities took. And then the last piece is the investors, who people have talked about, in the VCs. And today you can walk from the TLO, so that's 255 Main Street, I think, past a bunch of VCs, past the Broad, past the KI, past the Whitehead, all the way to the incubators where the startups form. And that's a 7-minute walk. So I think it was all this sort of surround sound that was the accelerator, at least for me, starting in the 90s.
Sangeeta, I want to see if you or maybe Phil has a sense of, we've kind of heard from David that the creation of the cluster was somewhat accidental, but I'd be interested in knowing if you guys are part of a planning for the future of it. Like what is the plan looking forward to weave entrepreneurship and the advance of molecular biology together, in the coming years? Is there sort of a 5-year plan for Kendall, or is it just allowed to continue organically?
Well, there are probably 50 5-year plans at Kendall.
Okay.
But one of them is referred to at the end of the film, and that is the convergence of high-power data, machine learning, and biology. And it's Google appearing in Kendall Square, and other Microsoft and many other companies moving into medical data and moving into genomic data and data processing, and a tremendous department of computational science at MIT and other places. Harvard is seeing this as well. So there's a synthesis going on that I think will have an amazing impact on medicine and how medicine is practiced. And that's something that many institutes are actually structuring their science around and wanting to make that happen. But maybe Sangeeta has other comments about her 5-year plan.
Do you have a startup company that's a bit of a synthesis company, isn't it? Does sensing maybe of cancer from the breath, is that it?
Yeah, I have a startup called Glimpse Bio, which makes synthetic biomarkers. So uses nanotechnology to create biomarkers in your body that didn't exist before for cancer and other diseases. And I think it's a good example of an engineering technology with medical applications. And I think when I started that company and I was getting it invested in, the very first thing I did was walk around my floor. And Phil is my across the street neighbor on the 4th floor at the KI. And we talked about who were the right investors. And then I went upstairs and I talked to Bob Langer, and then I showed the early pitch to Eric Lander. And that is another sort of accelerator, is the people that are around us and the connectivity. And they could tell me, Phil, I remember a very early conversation with you about Glimpse, and Harvey more recently, my new company, where you could say to me, well, this is the exciting piece. This is a piece that's never been done. You can't read that anywhere, right? Those are conversations amongst colleagues. So I think that's a really special part.
I want to just make sure we touch on the Asilomar conference. The movie is not just cures, it's also controversy. And just like the science, the controversy back then is sort of the same one as it is now. We have new capabilities all the time in biotech, and people fear that those are dangerous. So Joe, why did you include Asilomar in the movie? Of course, it occurred in California, so on the other cluster. What was your thought about putting that as a centerpiece of the film? And let our audience know, give us a synopsis of what Asilomar was.
Yeah, well, I mean, the folks here, several of them were there. David and Phil, I believe you were both there, so they can speak to it far more than I can. It was clear, that it was a turning point, and I remember in one of my early discussions with Phil saying, just how important that was, and to you personally, I think, and professionally. And what a turning point it was. I mean, it was clear. It stood out, and as a sort of a real milestone. And then, of course, it was what came out of it was the guidelines that kind of came out of Asilomar that then the city of Cambridge, once they were adopted. The city of Cambridge sort of said, no, hold on. I mean, that might have been okay for everybody else, but these guidelines may not be good enough, for us here in the city of Cambridge. We still have some concerns. So it was just this major turning point. But I think David and Phil, you were both there, right?
Yes, but there's a big difference. David was one of the organizers of that conference and was one of the leaders in the biomedical community or research community in the country that helped formulate a letter to the National Academy calling for the moratorium, and then worked to bring together the Asilomar conference that then had this enormous impact. So I just would love to take this opportunity to say to David, thank you. There were times I didn't agree with you, but in hindsight, I clearly need to thank you for taking that leadership at an MIT level and a national level. But you should comment.
Yeah, we really decided, and we means Paul Berg and Maxine Singer, others, that if we didn't take this bull by the horns, make it our problem, and guide its evolution and ultimately its resolution, that nobody else was going to do it. And it was going to end up in chaos. It was going to end up in a whole lot of special interests fighting with each other, and with no one really talking for the science. And we quite honestly did not know what we were getting into because there were too many questions that were unanswered and that no one had ever bothered to even think about the safety of the kind of work we were doing, about where it was going and what it might end up creating. And so we had to think all that through, and we had to tell our community, we had to ask our community, go slow. Wait, don't rush in. Let's see how this thing evolves. Because we have to be humble in the face of it. And there were a lot of people who disagreed with that. And Jim Watson being notable as somebody who said, we should just go ahead. Nature will take care of itself. And he had history on his side. Nature had always taken care of itself. But we weren't so sure this time. And we figured we will lose a couple of years of science that will make up in time, and did make up in time. But we'll gain the confidence of the rest of the world. And we did gain that confidence. And Cambridge was the microcosm of it. Because the mayor of Cambridge was a nutcase, but
David, that's not appropriate.
it's not appropriate. Okay, I take it back. But he served a very important purpose, which is that he reflected the skepticism that many people had about the ability of scientists to police themselves. And that's what we were saying we were going to do. And he did something really, in the end, brilliant, which was to set up a citizens committee. And that citizens committee made up of ordinary people, people who delivered your oil, people who, nurses, people without a whole lot of education in science, some with absolutely none at all. And he said, you people advise me. Do these folks know what they're doing? Are the guidelines being established in Washington really providing for the safety of the ordinary citizen? And that group worked very hard and became incredibly knowledgeable. And they said in the end, yes, the guidelines that are being established in Washington are what we should follow, and we will be as safe as we possibly can. This is science that has to go forward. People like David Nathan, who Harvey mentioned just before, a gray-beard physician at that point, said, look, this is the science that's going to create the medicine of the future. It has to be done. And they agreed. And it was their agreement that allowed things to move forward in Cambridge. It's a history which, I must say, I've never seen written in the form in which I lived it, and I believe it had its impact.
I think we should bring the conversation up to the present, and Asilomar is a perfect place to make the leap, because recently we've had, the CRISPR babies in China, where there was also attempts to replicate Asilomar at some level, to call or not call for a moratorium, to ask for a pause. I want to hear from you, David, on that, but I also want to ask Nancy Hopkins about her feelings about the editing of, children, the germline, the birth of genetically modified people. I mean, you were kind of in it at the beginning with James Watson. Did you anticipate that would happen? What was your feeling when you learned about it, and what's your feeling now? Is it an inevitable step? Where are you on this?
Well, I haven't been involved in the recent round of it, but this is a replay of something we went through this many years ago. And I thought it had been settled at that time. It's been revisited now that you can actually do it, more easily. But at that time, I think everybody felt that germline editing changing the germline was a really bad idea. Changing the germline of a human was a really bad idea. Changing, editing cells and stem cells and putting back, for treatment of diseases was a terrific idea. I don't think anybody was opposed to that, but the idea of changing the germline. And of course, at the time, we certainly didn't know enough to just start doing that. The question is, will we ever know enough biology so that you'd feel comfortable to go in and change the germline? And I haven't been involved in the recent debates about it. And I think there's probably people here who would be more up to date than I am about it. I still can't imagine doing it now. I don't think the thing in China should have been done. And I would be interested in what, I think, David, you've probably been involved in the recent debates. I have not, so.
There is the nature will take care of itself counter argument, though, that the genome itself is resilient, and so there's some leeway for.
Yeah, but for that particular individual person
Yeah.
is the problem, I think.
Right.
If you're doing the editing cells and putting them back, I think this is fantastic, you know, the potential. But for germline, I don't know. I can't see it yet.
David, do you see that process of debating germline editing being successfully concluded in the way that the recombinant DNA debate was? Is it being successfully, are we on a successful path or are we on a failed path?
I think this is a complicated area, and I don't want to be dogmatic about it, but I do think that there are certain kinds of editing of a newborn that are so positive for the future of that child that they should be done. And there are not many of them, and it's not a big deal statistically. It's not going to change gene frequencies or anything else. But Huntington's disease and lots of other single gene defects, if we could get them out of the lineage of our children, my children, other people's children, Harvey's child, I think that would be a good thing. And I don't see the downside of it, because all you'll really be doing is changing a gene from mutated back to the standard configuration that almost everybody else has. So we're not actually creating any new genes in the process. We're just getting rid of genes that have known deleterious effects. And anybody else is welcome to come in, and Harvey thinks about this probably more than I do.
I think it's unnecessary. There are other ways that are actually in practice that accomplish the same end. And as more and more DNA sequencing of people come online, people will know what genetic defects they carry. And prenatal genetic diagnosis is now quite common. Certainly in the Orthodox Jewish community, where virtually every young person is screened, they've been able to eliminate a lot of these horrible genetic disorders simply by either preventing or asking people not to have children, or doing DNA on the embryos and implanting only the embryos that don't have the mutation. So I think when you talk about germline editing, you have to ask, is it the only way of accomplishing it? And I would argue there are many easier ways that we can do it, and they can be done right now. And ethically, they're much easier than germline editing.
I would agree that that is the nub of the issue. I don't agree that it completely obviates the need.
Well, yeah.
I just would say, and I'm more conservative than David here, I've never seen a medical procedure practiced without experimentation that leads to it being effective and done right. And I just am very hesitant for the community to start experimenting in this domain, because there's individuals involved. And an individual is an important thing.
Kendall Square, figures in Kendall Square were not very closely involved in the episode, but the tools literally were developed here. An optimized CRISPR to work in a human cell, maybe even the cDNA, a license. So there's no CRISPR baby company in Cambridge that I know about. If there was, I would write an article about it. But it's interesting that more people here, I think, and in general, want to forego this, even though it springs directly from the technology. And interestingly enough, when I needed to find out about it, there was lots of people I could visit in Kendall, including working on monkeys and different things, who knew exactly, what the technical paths were, and that was very interesting. We're getting lots of questions about the commercialization of technology. Jeff Miller asks, he brings up Jonas Salk, how could you patent the sun? And this concept that the research at MIT is funded in large part by the government. So the question is, why should, given the public investment and subsidies, why shouldn't these treatments be publicly owned? And I'll give the question my own twist, which is that I calculated for somebody, not at MIT, at Harvard, but similar, a gene editing person, and they'd started a company based on a particular invention, and there was a lot of equity involved. And I figured maybe this person has, 1 day a week to work with a company, and I calculated the hourly pay, and it was $293,000 an hour for every Friday that had passed between the publication of the paper and the formation of the company. And that's pretty good pay. That's pretty good pay, better than an NBA player or a Hollywood star. So I think it came up in the film about the 60s and the 70s and the starting of biotech. And I think it's still relevant. Someone comes up with an invention in the lab paid for by the public, and then becomes also immediately the financial beneficiary of that from the license from the university. So, I mean, are we, I don't know, maybe Phil or Sangeeta, you can weigh in. I mean, is there a kind of a profiteering that goes on that is helpful to get things advanced, or?
Well, most of the science that, or a lot of the science that biotech is involved in it has its roots in basic science, frequently directly in basic science. But to take that basic science and to move it into a human therapeutic is a very complicated and long pathway. An example of this was RNAi, where the discovery was made in '98. The chemistry was worked out in around 2002. To take that to the first approved human treatment was 2018. It required several $billion. Clinical trials didn't work. New technology had to be obtained. New people had to be recruited. Various ways of putting together manufacturing of a new type of agent, learning how to use it safely, and improve. You could actually benefit patients was an enormous pathway. So the investment by the federal government in the foundation of this science leads to a lot of opportunities. One of which is the opportunity to take the new technology and make it useful. It's not different than chemistry, but chemistry has been fundamentally supported publicly in the last 100 years. And it's the chemistry in which your clothes is made of, the materials are made of, and it's all got to be translated. And to get the investment to translate requires a patent so that one can hope, if it's successful, to recover that investment and to make a company that will perpetually create new opportunities. So my motivation is not that the income that comes with it, though it's been very generous. It's seeing the benefit of what we've done in science actually being used to help people and solve problems in society. I mean, and that's the motivation.
Harvey actually has a grandson, I guess, who's the beneficiary of a Genzyme drug.
Correct.
Yeah.
actually, he's the beneficiary of a federal law called the Orphan Drug Act, which is an act passed by Congress, I believe, 1983, specifically to encourage the development of drugs for rare diseases. That is, companies of all sorts want to tackle, heart disease, cancer, other diseases that affect millions of people. This act deals with diseases like Gaucher disease that affect fewer than 200,000 Americans. That's still a lot of people. And it was because of that act that Henry Termeer decided to focus Genzyme on drugs for a number of rare diseases, Gaucher being the first. And I was privileged to help in the development of the drug. I had no idea that Gaucher disease was in my family. And it was only 10 years after the drug was approved that our daughter, having become pregnant, was offered prenatal testing, and it was clear that she and her husband were carriers of the disease. And the child she was carrying had a 1 in 4 chance of having Gaucher disease. And it's a difficult story to tell, but they had to make a difficult decision. And the fact that the drug was available to treat the disease factored in that decision. And as you saw in the film, Andrew's going to be 18 in November. And well, as I say, he's normal. He's actually well above normal, but that's a separate issue. The fact is, this act has brought great relief to many, many families with children with rare diseases. And we're now able to tackle diseases like sickle cell disease. There are gene therapies for that, which devastates many people in the African community, Blacks in the United States. We're able to treat it now. And as Phil said, it's costing a huge amount of money to take the research out of the lab, in this case, mostly out of Children's Hospital, into the clinic. And it's going to cost a lot of money for the therapy itself. But this is a decision society has to make as to whether we can afford treatments, not just for the cancers, but for diseases that may affect only a few thousand people.
The latest ones cost, the gene therapies, $2 million.
Yeah, but again, that's a lifetime treatment, hopefully. We don't know that for sure, but it has the potential to last for a lifetime. The gene therapies for the immune system, for the blood forming systems, almost certainly are going to last a lifetime. And when you factor that to the cost of hospitalization and treatment of these people over a lifetime, which is often shortened, together with the pain and the agony of the families that are treating these, we have to decide as a society that we can afford to develop drugs for these diseases, which are of course going to cost more because the development cost has to be covered by relatively few patients. But yeah.
Before we end, there's a couple more controversial points I want to touch on. And one is the question of diversity. And I think Nancy, we talked a little bit before the call, you may be working on it. And there's another documentary out there about the role of women in biotech and in molecular biology. So I took the opportunity to look at the MIT biology faculty page, and that diversity that people are talking about is, for the most part, well, in part, it's missing. I mean, there are women on the faculty. I don't think I saw, a native-born Black American on the faculty or Latin. So I just wonder, since Kendall kind of, it is this engine that's powering biotech, what are people thinking about this issue of, representing society as a whole in the makeup of the faculty? and also the financial beneficiaries.
Well, I mean, this is an issue I have worked on a lot in women in science, but I worked on it within the university, and a lot of progress was made in universities from practically no women when I arrived to, although in biology there were some, to in the biology department today, there'd be a quarter of the department would be women, and that would
Right.
be pretty standard. So I'm going to talk just about women and not about the other aspects of diversity. But a very unfortunate thing was that this increase in number of participation of women in science, which has occurred, particularly in the field of biology, did not occur in the biotechnology industry, which was developing parallel to what was changing in the university. That change did not happen in the biotech industry at the level of faculty. So for example, just to give one fact, you looked at who's in the biology department. That department has founded a total of 56 biotech companies, of which 2 were founded by women.
Mm.
So that gives you essentially women did not participate at that level, period. And that's why Sangeeta mentioned earlier on, some of us, Harvey has been fantastic. Sangeeta is a leader in this area. Susan Hockfield has been involved. We've been addressing the question of why, and it's a fascinating sort of question. And basically, I think what drove diversification of the faculty in terms of women in the universities really was civil rights, affirmative action, the women's movement. Those things did not touch the venture capital business world, basically. So these two things, one, we're going at different rates. And so unfortunately, when the biotech industry was developing, women just did not participate, really could not. And it's still a barrier, and but not so much for young women. It's beginning to shift, and it's much more, and by the way, the same thing has been seen at Stanford and so forth. So this is not a East Coast problem, this is a general problem. But fortunately, women in engineering do start companies, which Sangeeta discovered. So there was this contrast between the sciences and the engineering fields. And the same thing was found at Stanford, same thing. So in sciences, the women didn't. But even within engineering, we found that women have a much harder time raising comparable amounts of money. So it's a serious problem. And it's now being tackled by these pioneers here who are going to take this problem on and try to address it.
Well, hopefully, yeah, the next, I think the next movie in a couple of decades, hopefully will show a different picture. The effort that Nancy's describing is something we call the Future Founders Initiative to diversify the founding, mostly focusing first on women, but also including minorities, and trying to tackle that in a number of ways. So Harvey and I are launching on September 3rd, something we're calling the Future Founder Boot Camp, which is a series of, we hope, inspirational stories about academic founders, many of them women, but Harvey will also be speaking, so many inspirational figures. And it will be about starting companies and staying a professor. What does that look like? How do you do it? Who do you talk to? What are the nuts and bolts of it? And importantly, I think to the question from your audience, why would you do that? How does it change how you think about science and medicine? How does it change you as an inventor? How does it change your lab? How does it change the opportunities for your trainees? Why do we do this? And so that's just one thing we're doing. We also have a pledge from some of the VCs in town to diversify their boards, to give women more exposure to the industry. And MIT is also starting something like a sabbatical program to allow women to be entrepreneurs in residence at VC firms to learn the business. So those are just some of the ways that we're thinking about inventing the next chapter for Kendall Square.
Is the obstacle as simple as a kind of old boys network and deals made on golf courses? I mean, what is the barrier? How have you experienced the barrier?
Yeah, I think it's interesting. Nancy alluded to this. So I'm an engineer, and I'm the daughter of a serial entrepreneur. And I actually wonder sometimes why I have become a serial entrepreneur myself. And so part of it was cultural. I grew up that way. Part of it was my early mentors. Mm-hmm. My PhD mentor actually had me file a patent the night of my thesis defense.
Okay.
Part of it was colleagues who had me co-found companies with them so that I could learn. So I think I experienced many things that many women don't, that we're trying
Right.
to open the doors for them. But just coming to I think the crux of the issue is access to capital. So 2.7% of venture dollars in the US go into women-founded companies. So that is really a bottleneck that we need to crack.
Let's catch up with the pandemic. Again, we're here on Zoom. We're not actually in Kendall Square. What do people think? Oh, I mean, I heard you were actually tested. You lined up at MIT to get tested. What do you think the pandemic holds in store for this cluster? Are things just going to be? Are the labs open again? Are people going back to work? Are still going to be able to meet and have those serendipitous conversations? Or
for the biotech companies that I'm familiar with in Cambridge have turned to as remote engagement as can be usefully used to reduce people's densities in laboratories. And then in the research laboratory they had to manipulate things, instituted policies to give separation and protection and masks and records of entry and departure, and very intense testing. It's wonderful that the Broad Institute has really stepped up to the plate and expanded their testing capacity. It's a high quality, excellent institute and excellent tests. So, and most of the biotech, or many of them, are using that as a monitoring of the infections rates in their facilities. So COVID's going to change us. There's no question about that. And there will be more remote working. There will be more ways of actually engaging each other at a distance without travel and other activities, probably for the long-term benefit. But it's cost us a large 6 months of reduced activity now. And for me, I really see the long-term impact of this, not only just being COVID, but students who haven't been educated, people who haven't had opportunities to do breakthrough experiments that have been delayed for a year and will change careers. And economically, this is not going to be something that will end this fall. It's a readjustment economically. It's going to have long legs, many years.
right, we have questions from the audience about what the role of Kendall can be in sort of coming up with technology to counter the pandemic. And we just heard from Phil that the Broad Institute ramped up a testing pipeline that's being used locally, at least, and I think around Massachusetts and the Northeast. I've spent some time thinking, a lot of the vaccines that are coming out. I mean, it is the speed at which it's being done. It is a kind of incredible demonstration of the power of biotech at the current moment. I mean, it is amazing, and I'm just wondering if people have a perspective on how we may look back and do you think that biotech can answer this? Will be the sector that answers this
without question
challenge? Yeah.
We have Moderna that grew up in Cambridge. It's one of the leading companies producing or working on a vaccine with RNA. The whole genetic vaccination with a variety of different technologies is a product of biotechnology, much of which is developed at Cambridge and being developed. Monoclonal antibodies, a company that I'm on the board of, Vir, which is located in San Francisco, is got a remarkable set of high affinity monoclonal antibodies that will be going into patients soon, if not already, to treat the progression of and protection against COVID. So, I mean, and big companies and small companies are making enormous investments as rapidly as possible to impact on this. So if this virus can be controlled, it'll be controlled from that technology.
Mm-hmm. No, also diagnostics. I mean, many of the rapid, very ultra-sensitive nucleic acid diagnostics for COVID, antigen diagnostics, dipsticks, home diagnostics and so forth. They're all being developed around Kendall Square. So it's much more than just vaccines.
Right. Do people have ready to make odds on when a vaccine could become available? I mean, this is a hot subject, probably by November 3rd, apparently. But, I was just reading an article today that the Chinese have started distributing a vaccine and they have the winter in mind, and they want to vaccinate people to try and forestall kind of a crisis of COVID-19 during the colder months. And I thought that maybe was a reasonable thing to do. Maybe the risk balance is actually in favor of that idea. Although you often hear people saying, no, not until all the data's in, make a move. So I don't know who wants to jump in, but do people have a view about whether vaccine should be accelerated, perhaps even beyond, not waiting for the full data set? And when do you think that could happen by?
I'll give you just my feeling about this, which is that a vaccine generally doesn't work the first time you try it. You need to package it differently. You need to give it differently. You need to do something. If coronavirus is easy enough to vaccinate against, that we don't have to deal with that kind of surprise, then we will have a vaccine by the end of the year. But if it's not so simple, if we have to make tweaks to get it to really work well, if a lot of people who take the vaccine are not protected, then we have a wholly different story. And until we've done the testing, we don't know what the story is. We don't know what we're facing. And Moderna's doing a terrific job. There are maybe 6 or 8 other companies that are doing a terrific job of getting there. They've set up 30,000 patients, 15,000 getting the vaccine, 15,000 getting a placebo, and we'll look at the rates of infection. Now, an interesting question is, what do you actually look at? Do you look at the number of people who die? Do you look at the number of people who get very sick? Do you look at the number of people who get sick at all? Do you look at the number of people who get positive virus tests? What's the cutoff? What's your criterion? What are you really trying to accomplish? I actually haven't seen a good description of that. Now, it may be that the companies are keeping it to themselves and don't want to set up criteria before they've seen results. But that's not how an ordinary vaccine would be made. You'd establish those criteria before you injected the first patient.
The other thing with vaccines is, if you're talking about it in terms of a big population, is that people have to have confidence in the vaccine to be willing to take it. And we will have doubts about that.
Yeah.
The second is that an older population, such as myself, the vaccines do not work nearly as well as for younger populations who have more healthy immune systems. And therefore, the at-risk population, even with a vaccine, unless it's developed to be unusually potent in that population, will still not solve many of the issues of fatalities with COVID. So we'll have to have a combination both of vaccines and therapies that can manage this virus.
According to Joe, we cannot have a cure or a vaccine without a controversy. So I'm going to end on the controversy. I do think it's an interesting question, and a reader has asked it. There is a so-called conspiracy theory that the SARS-2 coronavirus was generated or otherwise accidentally released by this virology lab in Wuhan. Now, I think it's a conspiracy theory to believe it's true since there's not any evidence, but it's also, I think, scientifically perfectly possible that something like that could occur if you're working with dangerous viruses and changing them and using the very techniques that led to Asilomar, right, to make viruses more virulent or to test their capabilities. So I just want to know, what people make of that theory, which, if true, would split the edifice of science from top to bottom. I think some of the public says, scientists don't want to talk about this because if it were true, it would be such a terrible result of research.
It's incredibly unlikely that any laboratory would create a virus that had the subtleties and complexities that this virus apparently has in spreading in populations and being both rapidly spreading and have a significant lethality. No one could have anticipated that, in my opinion. So I think it's highly unlikely, and at some stage, as we get more and more sequences and we learn more and more about the ecology of the virus, we will have more answers about that. But David would probably be the most informed here. So David, what do you think?
I completely agree that it's very unlikely that it was engineered in a laboratory and got out of a laboratory. There's no evidence for it. We didn't know enough about it. It happens to be a class of viruses about which we know relatively little because there haven't been many cases of epidemics of it. And even in the MERS and SARS cases, it lasted a short while and was gone as a problem. I think it is possible that the virus was first evident in Wuhan, and it could have gone into a laboratory that was providing surveillance for the markets where exotic animals were being sold. We don't know anything more about that because what they did was to close those markets and clean them up instantly. And so there's no testing that's ever been done of what other animals might have had the virus, where the actual virus that was isolated came from. But there really is very little reason to believe that the virus was engineered. And what I look at is, do you see regions of sequence in the virus that look like they were artificially designed? And I think the answer to that is no.
The question of transparency is an interesting one. I suppose the next Kendall is not going to be, in Boston. It's going to be in Wuhan or Shenzhen or somewhere. And so we're going to have to understand how our cluster kind of communicates with theirs, and things are not going in a good direction in the current environment. I want to turn it back to Joe, whose film it is, and an excellent movie. I recommend everybody who hasn't watched it to watch it, with, your concluding thoughts with your gang here.
Well, thank you. And first of all, thank you all so much for participating in this and for participating in the film. Some of you, Harvey let me into, well, his family let me into his house to film with his family. So it was an extraordinary commitment that you all made, and I really appreciate it. I also want to thank the Coolidge Corner Theatre for hosting this session and showing the film. But I think listening to this fascinating conversation, I'm just struck by the history of Kendall is just endlessly fascinating, and still, obviously much more to be written. I mean, it was a place that at one time was landfill. It was marshland, and it was landfill, and then it became heavy industry, and then it's been lots of things over time. It was manufacturing and slaughterhouses and, robotics, and a lot of the, some of these more modern things are still going strong. But the idea that we can sit here and have a conversation for an hour and a half about modern Kendall and how it's and what the group here was at the foundation of, I think is extraordinary, and I can't wait to see what happens next. So that's over to Sangeeta.
Sangeeta, what's going to happen next?
We're going to have more women and minority founders.
Well, Joe ended the movie to be continued. And I think he just expressed why he put that at the end of the movie.
Oh, this is just the beginning, just the beginning. So thank you all very, very much.
Thank you.
Thank you.
Thank you.